So very good evening everyone. Students. So we will start from the yesterday's lecture that we have finished, the affinity chromatography. So this method relies on the knowledge of the properties of the enzymes that you want to isolate. So basically in this your antibodies are playing the role. So mainly the antibodies are the one that are playing the role here. So antibodies must be available that raised against the enzyme, and the enzyme must know to bind to some substrate such as ligand or non-covalently in the reversible way. So it is the most specific chromatographic method that relies on the specific binding of a target with its own specific ligand. So you have your close binding proteins that are attached to the glucose here on these beads, and then addition of glucose is then added to it. So these glucose resins to isolate glucose binding protein from conclavin A. Then glucose binding protein is released on the addition of the glucose further. We can see this also the same way like what we saw in earlier way of doing the chromatography. So we have loaded with the pH buffer, and we are having these antibodies inside them. So we will have a protein which could be recognized by the antibody in the red color, and the blue ones are the not recognized by the antibody. So once you wash them, you elute them with the pH buffer 3, so you can separate them between blue and the red one, and that's how you can have the different results. So there are some drawbacks to the affinity chromatography, that the binding to the ligand or antibody can often be strong, that very harsh, and needed to elute the enzyme, and this leads to denaturation. Then there's a non-specific binding may also occur, especially where large ligands such as antibodies are employed, and this non-specific binding can overwhelm the correct binding sites at very high protein concentrations, leading to the pure purification. So let's see how we
Separation in affinity chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and elution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. Non-binding molecules will pass through in the flow-through during washing with binding buffer. In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by change in the buffer composition. A common way is to increase pH of the buffer.
Now comes the next, that is your high performance, your high pressure liquid chromatography, which is the most enhanced version of all the chromatography that we have discussed. So in this, resin sizes are more refined, and all the chromatographic methods described above can be applied with the HPLC columns and equipment, but with the HPLC the separation of peaks is almost better. So basically in this you can see these peaks coming out. So 1, 2, 3, 4, 5. So this is absorbance versus the time here. So we see some peaks here with respect to some chemicals that you're looking for. So like the first chemical belong to something, 2 to something, 3 to 4, and different. And with along the time we will extract them over the column. So your columns are like C18 column here, so where the separations takes place. So this is from the advancing the chemical sciences from the Royal Science Committee. So let's see how it is done in reality. Maybe I should play outside, then it will be better.
HPLC stands for High Performance Liquid Chromatography, but could equally well stand for High Pressure Liquid Chromatography. It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase non-polar. In paper chromatography, the solvent moves along the paper by capillary reaction. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, a component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a nonpolar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
Yeah, so that was the peak thing. That's how we do all um, your HPLC in reality. So there is also fast performance liquid chromatography, which is a type of liquid chromatography where the solvent velocity is controlled by the pumps actually. So the pump controls the constant flow rate of the solvents, and they are assessed by through the tubing from an outside reservoir. And the flow rate of solvent is set through the computer input and controlled by the pumps only. Now comes your next part, the biggest part, that is your gel electrophoresis. So basically this is done to separate your proteins on the polyacrylamide gel, which forms a three-dimensional molecular sieve. So under the denaturing conditions, the gels can dissolve proteins of the size of from 10 to 250 kilodalton, and proteins have nucleic acids. um, Proteins and nucleic acids could be migrate over this electric field, and we can separate them according to size and charge both. So the most popular method is SDS Page, that is your sodium diadosyl sulfate polyacrylamide gel electrophoresis, where electrophoresis is coupled to a sieving device and that differentially retards the large proteins. So in this, basically, if you see, you have your gel will be running over the vertical point from negative to positive, and you put your protein, the mixture of a macromolecules here. So the smaller one will go faster and will be at the bottom over this porous gel, and then the green ones in the middle and the yellow ones at the top. So in this way, you can separate your all proteins. And the acrylamide that we're using, so it will make, what is it happening with the polyacrylamide gel is actually a gel is made by polymerizing the acrylamide and bisacrylamide. So you have a bisacrylamide and acrylamide, they get polymerized together to make this chain membrane structure, which makes through these pores then your protein will be going out of it. And your SDS, what is SDS doing? SDS will coat over that protein. It will give a negative charge to it so that your protein could move from negative to positive end. It will all cover your sodium dodecyl sulfate will cover it and will cover your protein with it. So that protein could be separated according to the different sizes. So that's how the page role and SDS role together, they are playing a very essential role at the end. So this SDS page staining is done like this. You can save them. So here we can see on the side is your like we did in the DNA fingerprinting. You have your molecular markers on the right, and in the center is your gel running on. Yeah, your gel is running. Your various protein samples results are here, and this is the data kept for the same for the autoradiograph. You can keep it for the future. There's some also another way with the different like coating results. So this is staining for 1 hour, for 1 hour, for 1 hour, both in three cases, and then destain with them timing 1 hour, 0, and this wash enhancement is increase in this. The same results. So here we can't see much better results, but here we can see clearly visible. And here crystal clear background and excellent sensitivity to 8 nanogram. So this seems to be the best method, the Pierce gel code blue stain reagent. So we can see very fine data as compared to the left one with this technique. So that was the basics behind the gel electrophoresis. Now let's talk about isoelectric focusing gel electrophoresis. That is your 2D gel electrophoresis. So before running your 2D gel electrophoresis, you run your samples according to their pH level. So we run, so there will be high pH and low pH provided to a strip, and your protein will be added over the strip. And then the high pH and low pH, what will happen, your protein will attach to a point where they have neutral pH, where the exact pH is all about, the red, blue, yellow and orange. So polyamphilites, small multi-charge polymers. So gel contains polyamphilites that form pH gradient in the gel under the electric field, and proteins can then separate it based on their pI values. So pH gradient polyamphilites with many pI values differ by 0.01. So this is how the in the in isoelectric focusing gel you get better results than the SDS page actually. So this is 18 kilodalton ELF5A protein. This is the unmodified and modified version. So we can see the modified version to be much better, much broader present. We can detect difference of 1 charge per molecule here. So let's see how we prepare the precipitation for this 2D gel electrophoresis, how mainly the precipitation is done for the same. So this will be without audio, this video. So this is the way we precipitate the protein. So everything will be given under the titles here, the down. Okay, so once you have precipitated your protein, everything is done over that isoelectric focusing strip. So you put them over the gel after the separating by charge. You put them to separate by their size now. So we will separate them over the SDS electrophoresis, like the one we have discussed before, and then we will see this kind of results. So once your proteins are separated according to the charge, then they will went for the separation over the um, yeah, according to their size. So you will see this pattern. This is a proteomics pattern. The 2D gel electrophoresis results. At the end, you will see all the proteins that are present in specific organisms, whether it's a healthy state or disease state. So you can differentiate between the various proteins, whether that is present or not, and you can make very clear, insightful, like from the diagnostic perspective, what should be the results out of it. So very essential and very good part of the field. So let's now have a look over the how it is done. 2D gel electrophoresis. So this is a complete 17 minutes video which professor will be telling about from the 2D gel electrophoresis, from running it over the pH and then running over the SDS gel electrophoresis. So all steps will be covered in them. So be patient. The professor who is telling from the BioRed, he is extremely genuinely very big person in his field and very expertised. So, and that's what we were doing for whole years. When we were running also 2D electrophoresis, is the same procedure has been shown by our this esteemed personality. So have a look then.
Hi, I'm Steve Freeby. I am a staff scientist at Biorad Labs. In this video, I am going to demonstrate how to run good reproducible 2D gels. The video is going to be broken into 3 segments. The first segment will be how to apply, sample, and rehydrate the Ipg strip. The second segment will be on how to equilibrate the Ipg strips in preparation for running on the second dimension gel. And the third segment will be staining of the second dimension gel. In this video, I hope that you get tips and techniques and tricks that will help you run good quality 2-dimension gels. After sample preparation, the first step to 2D is to rehydrate the Ipg strip. And I'm going to demonstrate this process using Biorad's 2D Starter Kit. The 2D Starter Kit is a great tool for learning 2D gel electrophoresis. It contains all the reagents and an E. coli sample for running a 2D gel. The first step is to prepare the sample and the reagents according to the instructions in the manual. The components of the rehydration buffer are urea, CHAPS, DTT, ampholyte, and bromophenol blue. I will be loading 200 micrograms of E. coli sample that has been reconstituted in rehydration buffer onto an 11 centimeter pH 4 to 7 Ipg strip. To rehydrate the strip, I will load 200 microliters of sample into the focusing tray in each lane that I'll be running a strip. I am using a focusing tray that is specific for 11 centimeter strips. Trays are available for all Ipg strip lengths. Tilt the tray toward you to allow for even distribution of the strip. An easy way to do this is to take the focusing tray, put it on the edge of the lid to the focusing tray, and you have the tray at an angle towards you. Dispense sample along one edge of the lane of the tray. And you want to dispense it in a nice even bead from one electrode across to the other. One even bead. I have shown you this with the focusing tray, but this can also be done with the rehydration tray. Now we are ready to place the Ipg strip into the focusing tray. An Ipg strip has 2 sides. One is a plastic protective backing and the other is a dried gel. You begin by removing the plastic protective backing with forceps, and you can tell where that plastic protective backing is by a slight indent on one side of the plastic pieces
the side that has indented is your protective backing. You pull that rapidly across, and now you have gel side up. Take your forceps, hold onto the strip on the negative side. You'll see the other side has a positive side. The negative side is the opposite of that. And you can always tell when the IPG strip is facing down, but you'll be able to read the numbers of the pH range of the IPG strip in the correct orientation. Now the gel will be loaded onto the sample bead, gel side down. If you are running multiple samples, I recommend labeling your strip before the step. Make sure to label the side of the gel and not the protective backing. To load the IPG strip into the focusing tray, place the positive end of the IPG strip into the positive electrode of the focusing tray and slowly drop your IPG strip into the lane of the focusing tray. Being very careful not to trap any air bubbles. If there are any air bubbles trapped, you can pick up the end and gently reposition the IPG strip until the air bubbles are expelled. If you don't remove air bubbles, there will be areas in the IPG strip that do not rehydrate properly, and that will cause serious anomalies in your first dimension isoelectric focusing run. The initial step of the first dimension separation is to rehydrate the strip. I will do passive rehydration in the focusing tray. Other forms of rehydration are active, which may be used for high molecular weight proteins, and cup loading, which is ideal for the separation of proteins that will be separated at extreme basic and acidic pH. Place the focusing tray into the protein IEF cell, making sure to align the positive electrode of the focusing tray with the positive electrode of the protein IEF cell, and add enough mineral oil to cover the strip in each lane. The purpose of the mineral oil is to prevent the strips from drying out during the course of the rehydration and the run. Now I am going to program the protein IEF cell. Instructions for programming are provided in the protein IEF cell manual. Typical rehydration times are 12 hours and focusing time ranges from 6 to 8 hours. At the completion of focusing, if you do not have time to run the second dimension gel, first remove the strips from the focusing tray, blot off the excess mineral oil, and place the strips into a rehydration tray at -80 degrees C. The steps following rehydration and focusing of the IPG strip are equilibration of the IPG strip in preparation for the second dimension gel, which I will now demonstrate. The equilibration process is a two-step procedure. Equilibration buffer number 1 contains DTT for reduction of proteins in the strip. Equilibration buffer number 2 contains iodoacetamide for alkylation of proteins in the strip. Each solution contains SDS for denaturation of proteins in the IPG strip to ensure optimal performance in the second dimension. Prior to equilibration, I will remove the IPG strip from the focusing tray and blot the excess oil off of the strip. First, pick up one end of the IPG strip, turn it over so the gel side is up, and place the IPG strip gel side up on a dry piece of filter paper. Wet another piece of filter paper and place the wet filter paper onto the IPG strip and gently blot the excess mineral oil off the strip. Be careful not to press too hard to damage the IPG strip gel. The next step is to equilibrate the strips. I prepared buffers from the Ready Prep 2D starter kit as instructed in the manual. Remove the wet filter paper from the top of the IPG strip. Place the IPG strip into a lane gel side up of the rehydration tray. Add 2 milliliters of equilibration buffer number 1 to each lane of the rehydration tray that you have a strip. place the rehydration tray onto a shaker rocker table for 15 minutes. Note that the rehydration tray has an angled end and a straight end. Whenever you're pouring out liquids, use the straight end so that it does not slide out of the tray. Now add 2 milliliters of buffer number 2. Shake for 15 minutes and pour out. At this point, the strips are ready to run on the second dimension gel. I am going to use Biorad's Criterion precast gels, which accommodate the 11 centimeter IPG strips that we ran earlier. Prepare the gel by removing the sealing tape from the bottom of the gel and slide out the comb from the top. Rinse the wells with good quality DI water. Tap the water out into a beaker and then place the gel sideways and remove with a piece of filter paper the excess water from the wells. Be careful not to touch the bottom of the gel as it can damage the surface of the second dimension gel. We usually do this because unpolymerized acrylamide can cause anomalies in your second dimension gel if it's not removed. To remove the strip from the tray, place the forceps on the tray and gently push on the plastic backing of the gel to push the strip towards the angled edge. Lift up the strip from the right tab. I am going to rinse the strip off in running buffer that I have previously prepared. Dip 2 or 3 times. This will remove equilibration buffer number 1 and 2 that may still be on the surface of the IPG strip. Place the IPG strip gel side up. Place the plastic backing against the back of the cassette. Position the strip centered in the middle of the well with your forceps and gently push on the strip until it touches the gel. And you can tell when it's touching because you'll feel a little bit of springiness, a little bit of resistance. Don't push too hard. Now I will insert a Precision Plus standard plug, which is a broad range standard that has been embedded in agarose, into the reference well on the right side of the gel. Break off one plug, hold onto the handle and break off the spatula, and using the spatula, push out the agarose plug. For Criterion gels, I usually cut the plug into 4 slices to match the size of the well. In this case, I need 1. With forceps, take one of the slices, place on the back of the gel cassette over the reference well, and gently push the plug into the reference well. At this point, we want to seal the IPG strip and standard gel plug into the well of the gel. I will use Protein Plus Overlay Agarose. Heat in the microwave oven until it boils, and when the temperature of the agarose is no longer hot to the touch, take the hot agarose and fill up the well. And do this in one rapid action, side to side. Working quickly, use your forceps to push the IPG strip against the gel to push out any air bubbles. Agarose is going to solidify fairly fast. You can see the agarose filling in the place of the bubbles as they get pushed to the top. Small air bubbles are not an issue. Try to get some of your larger ones. and you should not have any problems for your second dimension gel. Now that the agarose is solidified, I'm ready to run the gel. In this case, I'm going to run the gel in the Criterion gel box, which holds 2 gels. In most cases, you're going to run multiple gels and you want to use a Criterion dodecacell. Remove the gel cassette and place it into the Criterion cell. Load the upper buffer chamber with running buffer. Place the lid onto the Criterion cell. Insert the leads into your power pack power supply. Program for 200 volts for 1 hour. Constant voltage. And start. The second dimension gel has now completed running. You know it is finished when the dye front has reached the bottom of the gel. We're now going to remove the gel from the Criterion cassette and prepare the gel for staining. Remove the gel from the gel box, pour out the upper buffer, and open the gel by using forceps or a similar tool by pushing the forceps against the plastic cassette towards you at the top and work your way down. So fold open like so. After I open the gel cassette, I use forceps to remove the Ipg strip from the surface of the gel. Starting at the bottom of the gel, gently pull the gel away from the surface of the cassette. The easiest way to do this, turn the gel upside down over the surface of the staining solution, remove a corner, and remove a corner until the gel falls into the solution. Biorad offers many different staining choices for 2D gels, ranging from the visual stains, the traditional Kamasis and silver stains, to the fluorescent stains, flamingo and cypro. In this case, I am staining this gel with BioSafe Kamasi, and the first step requires 3 10-minute rinses in water to remove SDS from the gel. We are now going to stain the gel using Biorad's BioSafe Kamasi stain. Following the 3 10-minute water washes, we're going to transfer the gel to a tray of BioSafe Kamasi, and we are going to stain the gel for 45 minutes to 1 hour. Following staining, we are going to transfer the gel from BioSafe Kamasi back into water, and we are going to destain until you get desired background. At that point, the gel will be ready for imaging. Here you can see that I have generated a gel with a clear background and well-resolved spots. I have now completed the entire process for running a 2D gel. I hope that the techniques and tips will help you when you run your next 2D gel. As you have seen, Clean Sample and the Biorad 2D Starter Kit will help you generate good quality reproducible 2D gels. For additional information and techniques and tips on sample preparation and 2D gel electrophoresis, you can visit our Expression Proteomics website at www.expressionproteomics.com.
Okay. Seven percent. So this is the results that we get from all the different chromatography, from ion exchange, from molecular exclusion, gel exclusion chromatography, and affinity chromatography. So single page can be accepted as a criteria for pure protein. So if we get a single band, that means the protein that we get is the pure one, is the best one, and we can use it for further analysis. So further, the next part, you have done the isolation, you have done the quantification, you have done the purification with various techniques of chromatography, HPLC, 2D gel electrophoresis. Yeah, all things are done now. Now it's time to the mass spectrometry in order to analyze your protein, in order to find out the how many amino acids is your protein is made up of, what is the sequence of your amino acids. So you take that protein and you run over the mass spectrometry, and it will gives you very instantly results about how many of your sequence of a protein it is made, and how much valuable is your data at the end. So in this, basically in the MALDI-TOF, this is a MALDI-TOF spectrometry, whereas the MALDI means matrix assisted laser desorption ionization, whereas TOF is the time of light. So in this, a laser beam is ionized and over the protein in the matrix. So this is your matrix. Yeah, on the matrix your protein is added. So ionized proteins accelerated towards a detector, and the detector monitors the arrival of ions as the laser triggers the clock. So protein samples are ionized here, and then afterwards uh, it's being, yeah. So these laser then on over the protein, this laser is coming from this laser light. After the beam splitter, it goes to the prism and then hit over the protein. Then it will goes to the electric field which will accelerate them. So your positive ions which are bigger will become smaller and smaller, and then we detect them, and then it goes to the transient recorder, and we can see the results at the end. So we see this kind of results in the MALDI-TOF with various ions to be present. So like for insulin, there is present, there is a beta lactoglobulin is present. So insulin, the value of the intensity according to the mass to charge ratio seems to be 5733, whereas for the beta lactoglobulin it seems to be 18364. Yeah. So MALDI-TOF spectrophotometry looks like this, the various results of the same. Now let's check how the MALDI-TOF spectrometry is done in practical level.
Worker MALDI-TOF process. MALDI-TOF analysis provides additional structural information for target molecules. As MALDI-TOF is a two-phase process. Fragmentation can occur by application of higher laser energies, known as LID, laser-induced dissociation, or a background gas can be introduced to promote ion collisions. This process is known as Collision-Induced Dissociation, or CID. Samples are co-crystallized with a suitable matrix on a sample carrier. The samples are vaporized by the laser into a vacuum while being ionized at the same time. During this process, the sample molecules can become excited. The now charged and excited particles are accelerated through the application of a high voltage, and fragmentation can occur as they travel through the flight tube. In this process, precursor ions and their respective fragments travel together at the same speed. Based on the expected time of flight of the precursor ions, specific ion packages may be individually selected, and all other ions blocked from continuing through the flight tube. The selected ions are then re-accelerated, with the precursor and fragment ions traveling at different speeds towards the reflector detector. The ions travel via the reflector towards the detector, and thus a TOF spectrum is recorded. The detection of the selected precursor ion and its fragments creates a TOF spectrum. Whether fragmentation occurs through the use of higher laser energies or a collision gas, the structural details derived from the MALDI-TOF spectrum provide a deeper analytical insight of the targeted compound.
So now one of the applications of this mass spectrometry and these techniques. Yeah. So this is the last video of today's session. Then we will be done for it. Video 20 minutes.
Today, we rely heavily on biotechnology for many of the necessities of life. For the food we eat, the clothes we wear, fuels for transportation, earth-friendly plastics, pharmaceuticals to treat our illnesses, and dietary supplements to help us stay healthy. Many of these products are produced using three core biotechnology processes: fermentation, recovery, and purification. Fermentation is basically cell farming. We program cells to produce a product. We nurture them as they grow and reproduce, and then we harvest them. In recovery, we separate our product from the cells where they were housed. And then in purification, we go in everything else that's contaminating our product, leaving us with a very pure, concentrated solution. In this program, we're going to look at a typical purification process used in the manufacture of GFP, green fluorescent protein. GFP is broadly used as a biological marker because it's very well tolerated by most cells and doesn't interfere with normal cellular function. We'll examine the technologies, equipment, and materials used, how to prepare for the process, and how the GFP purification process is managed, step by step. There are two main operations used within purification: chromatography and filtration. Before we can appreciate exactly what these process steps are accomplishing, we need to take a closer look at what our green fluorescent protein has been through so far. Back in recovery, our harvested cells from fermentation were homogenized. Our target protein was inside each cell, so to get at it, the cells had to be ruptured. Homogenization freed a flood of cellular components, including membrane debris, cytoplasm, DNA, and proteins, including our target protein, GFP, and they were all mixed into the buffering solution used to suspend the cells. Most of these solids were then removed through centrifugation, or by centrifuge, but the liquid that remained, the clarified lysate, was still rich in biological products, including dissolved chemicals, proteins, and other impurities. Amazingly, through purification, we can target a single protein within this biological soup. In our GFP purification process, we'll be using multiple types of column chromatography. The column is a cylinder filled with glass, ceramic, or polymeric beads, which are engineered to interact with or bind with molecules based on one or more physical properties. Chromatography relies on differences. Each molecule has a unique set of physical characteristics, such as size, charge, or extent of interaction with water. Chromatography uses these differences to separate the target protein from other proteins and chemicals. Sometimes size is used to differentiate. Some beads have small holes in them and can temporarily trap or at least slow down smaller molecules as they travel through the column of resin beads, while molecules too large to enter the pores move around the beads and exit the column first. This type of chromatography is called size exclusion. In the case of charge, opposites attract, so a negatively charged chromatography bead will attract and bind to positively charged components in the process stream. Likewise, a positively charged bead will bind negatively charged components in the process stream. This charge-based chromatography is called ion exchange chromatography. And then there's water. Molecules that readily interact with and dissolve in water are called hydrophilic, water-loving, while those that don't are called hydrophobic, water-hating. Proteins contain regions that are hydrophobic and regions that are hydrophilic. Because water tends to form a shield around the hydrophobic patches within the proteins, they are not exposed to interact with the resin beads. By adding salt to the protein solution, we remove the water shield, exposing these hydrophobic patches on the protein and resin so they can interact. This is how HIC, or hydrophobic interaction chromatography, works. In our process, which uses ion exchange and hydrophobic interaction chromatography, the chromatography equipment is housed on a skid to make it compact and mobile. The main part of the apparatus is a glass column filled with resin beads, but we also have pumps to move the clarified lysate through the process, a supply hose and port to feed the column, a pre-filter to remove any remaining particulates, usually solid cell debris that has not previously been removed, before the clarified lysate enters the column, an exit port for the processed solution, and auto switching valves for directing processed solution to either waste or collection. To help monitor the chromatography equipment and the solutions flowing through the unit during the process, a number of sensors are located along the product flow path. There is an electrical conductivity sensor at the column inlet, a pressure sensor just before the pre-filter to help identify a filter clog, a flow meter to measure the rate of solution movement through the column, and an air sensor to ensure that no air has entered the flow path. As a solution leaves the column, it passes a UV sensor that reads optical density, a second conductivity sensor, and a pH sensor that measures how acidic or basic the solution is. The conductivity sensors let us know when a new buffered solution has filled the column. When the conductivity reading on the exit of the column matches the reading from the sensor at the inlet of the column, then we know the new solution has completely displaced the old one. The ultraviolet UV sensor monitors the concentration of protein in the product by observing the optical density of the passing solution. This sensor works hand in hand with the valves on the exit of the column. Through the controller program, we can set a protein concentration threshold. When the optical density of the solution leaving the column is below the threshold, the valve directs the flow to waste. When the optical density of the solution leaving the column is at or above the threshold, this means that solution contains our purified product and the solution is directed to a collection vessel. TFF, Tangential Flow Filtration, is at its heart a simple process step. We're going to pump a fluid through and across a special type of filter known as an ultrafiltration membrane. The size of the pores in the filter material determines what passes through and what's held back. As with almost any filtering process, we can choose what we want to keep. The solution that passes through the membrane is referred to as the permeate. Because the pores of the ultrafiltration membrane are small enough to keep the product from passing through, the permeate contains no product and is sent to waste. The portion of the feed stream that does not permeate the membrane is called the retentate. It contains the retained product and is the stream we are most interested in. What makes TFF different is a core technology that enables it to be faster, more efficient, more flexible, and even self-cleaning. In conventional terminal filtration, a fluid is pumped directly into a filter. The particles within the stream that can't fit through the pores of the filter build up at the filter surface, eventually clogging it. In tangential flow filtration, the stream moves across the filter, that is, tangential to the filter, instead of directly at it. The cross-flow current actually picks material back out of the filter media or membrane and into the stream. This retained material, called retentate, is recirculated to the supply tank and will continue to loop through the filter for as long as the process runs. We'll be using TFF for two different tasks within the purification process: concentration and Dia filtration. We are processing purified GFP from a chromatography step. The fluid from the chromatography step is purified green fluorescent protein dissolved in a buffering solution. In Dia filtration, we add new buffer to the GFP retentate while displacing the old buffer, effectively exchanging buffer solutions. Note, the GFP is retained by the membrane. If we don't add a new buffer, then we're concentrating our solution. Concentration is simply the removal of water and buffer components from the feed solution that results in a more concentrated solution of green fluorescent protein. From operation to operation, or product to product, the purification process can look quite different. It could be as simple as a single chromatography step, one cycle of TFF to concentrate the product, and final filtration. Or it could involve several different types of chromatography, Dia filtration between chromatography steps, and a final conventional filtration. Our green fluorescent protein process is pretty typical of the process for biopharmaceutical products. The clarified lysate will be pre-filtered, go through anion exchange chromatography, and the GFP collected and pooled. Next, ammonium sulfate will be added to make the solution high salt, followed by hydrophobic interaction chromatography, HIC. The GFP is then collected and pooled again. The solution then goes through a TFF ultrafiltration step, which includes concentration of the GFP followed by Dia filtration to exchange buffer solutions and remove salt. And the process finishes up with final filtration into bulk bottles or bags. It's time to gather everything we'll need for the purification process and ensure that the area and equipment are ready to go. The chromatography skid and TFF system are checked for proper operation. Process hoses are attached and examined for leaks. We verify that we are using the correct column resin and that the resin is properly packed. The column is filled with a storage buffer, and the product path is checked for trapped air and purged if necessary. Our raw materials include the clarified lysate from the recovery process, various buffer solutions that are tailored to specific process steps, and ammonium sulfate that we add to one of the buffers to make it high salt. The purification process is managed through the use of a batch record. There is a separate batch record for each processing operation. These documents lead the operator through the process step by step, with each step requiring a sign-off and separate verification by a second operator. The batch record also includes spaces for documenting times, activities, operation steps, and instrument readings. Before the process can begin, the purification area must be cleaned, disinfected, and organized. Any unnecessary equipment or materials are removed. All equipment must be cleaned, sanitized, and set up as required by standard operating procedures. All required materials and documentation must be gathered and prepared before the process may begin. The purification process begins as the transfer tank of clarified lysate from the recovery process is connected to the inlet pump on the chromatography skid. The first chromatography step in our green fluorescent protein purification process is anion exchange. At this point in the process, the pH of the clarified lysate is about 8.0, which means that the protein is negatively charged. Because it is negatively charged, GFP will bind to the positively charged anion exchange resin. The pump draws the lysate from the vessel, past the first conductivity sensor and pressure sensor, and through the 0.45 micron pre-filter. The pre-filter removes any residual cell debris or other particulates that may have contaminated the solution. If the pre-filter begins to clog, the pressure sensor at the inlet side of the filter will register a rising pressure, and the controller will signal the need for a filter change. After pre-filtering and before the column, the lysate passes through a flow meter and an air sensor. Then, as the lysate passes over the resin beads, the negatively charged protein binds to the positively charged beads. The solution leaving the column passes a UV optical density sensor, a conductivity sensor, and a pH sensor. The optical density sensor's low readings confirm that the GFP is not in the solution, so the outlet valve sends the solution to waste. When all the lysate has entered the column, or when the capacity of the beads to bind the protein has been reached, it's time for elution. Elution is the release of, in this case, green fluorescent protein from the beads by using a new solution, in this case a buffer that includes NaCl, sodium chloride solution. As the new buffer is pumped through the beads, at some point the GFP no longer binds to the beads and is released into the buffer. The resulting product stream is usually referred to as the eluate. The UV optical density sensor, which measures protein concentration, indicates when product begins eluting from the column. At this point, outlet valves are switched to allow flow of the eluate, the product stream, to a collection vessel. When the UV sensor indicates that all of the GFP has come off of the chromatography resin, the outlet valves are switched to waste. When all of the eluate has been collected and pooled, the anion exchange chromatography step is finished, and it's time for HIC, Hydrophobic Interaction Chromatography. Hydrophobic interaction chromatography is based on the principle that hydrophobic chemicals on the resin surface will bind to hydrophobic patches on the GFP protein. In order for this to occur, the resin and protein eluate have to be in a high salt environment to remove the water shielding. The salt we use is ammonium sulfate. To remove the attached GFP protein from the HIC column, we simply lower the salt concentration during elution, causing the water shielding to reform, and the GFP protein detaches from the resin into the elution stream. The protein-rich eluate is collected and pooled. The product is now ready for the last major step, TFF. At this point in the process, tangential flow filtration will be used to concentrate and Dia filter the GFP product stream. The eluate is rich in green fluorescent protein, but it's still too dilute and too high in salt. As the solution moves through the TFF apparatus, it leaves the supply tank, is pulled through a pump, past a pressure sensor, and then across the filter membrane. Everything that passes through the membrane, including the buffer solution, is known as permeate and, for this process, is sent to waste. The GFP protein is larger than the pores of the membrane and therefore is retained. The retained material, called retentate, is recirculated to the supply tank. Recirculation of the feed continues until the desired concentration of GFP is achieved. Following concentration, and while the protein solution is recirculating, a new solution, a storage buffer, is added to the feed. In effect, the protein is being washed by the flow of a new buffer solution in and the old buffer solution out. As this Dia filtration step proceeds, the buffer solution that is being added to the feed replaces the buffer solution that the GFP was originally in, effectively removing any remaining salt as well. When this process is complete, the GFP solution is routed through a 0.22 micron final filter and then collected in appropriate containers, usually bottles or bags. The purification process is complete. The green fluorescent protein concentrate can now move downstream to final fill finish to be freeze-dried and packaged.
So that's it, students. I will share all these slides with you. I've already shared the PPT, and yeah, I will share the videos now with you later on. So thank you so much for today's lecture. We see us then tomorrow, same time, and we continue tomorrow with the, I think, dull electrophoresis. Yeah, we will continue more different types of electrophoresis tomorrow. Until that time, take care. Have a nice evening. Yeah, sleep well. Take care. Good night. Bye bye.